All right, let's begin. So, the point of this lesson is to talk about G proteins and olfaction, which is smell. And smell and taste, you've probably heard before, are definitely connected. Actually, smell is most of what you perceive as taste. And so, for each of these mechanisms, anytime you actually smell something in the world, it's because a little molecule from this, and so we've got nice little cilantros over here, which I think are delicious, but I know some people do not like. And actual molecules from this are coming and they're floating through the air, coming to your nose, binding to a G protein coupled receptor, activating a G protein, and starting a signaling cascade that leads to a pulse of electricity that's actually going to go and signal to the rest of the brain. So, one of the chemicals that you can find in coriander here, or cilantro, is going to be this sort of group of things: decanol, 2-decanol, and 2-undecanol, which is an extra carbon long. And these are compounds that we are going to perceive as somewhat soapy tasting. And yeah, there are some people that have inherited versions of the G protein coupled receptor that binds these that are going to signal a lot, and some people inherit versions that don't signal very much. And so, depending on that, it doesn't necessarily mean you're going to like it or not like it, but you're either going to get a very strong signal or a very weak signal from a particular stimulus. And then from that point, it's just a signal. It's either strong or weak. And your brain is going to figure out what it wants to do with that based on your entire life experience and everything else that's going on, and all the connections in your brain and ways that we don't really fully understand the big picture of, but have a pretty good picture of the small scale picture of. So, yeah, all this is going to happen. But before we go into this, I wanted to look at some of the underlying science here, because I think this makes for very interesting conversation. I'm hoping that you can have a wonderful Thanksgiving conversation with your family, if you're back with your family for Thanksgiving, with some of the stuff in here. And it's all good science, and most of it has the references on there, so you can go and look it up. All right, so first of all, who has the most odorant receptor genes? So each odorant receptor can bind to a narrow, fairly small family of molecules. And so to be able to smell more stuff, you have to have more receptors generally. And so if you look, we've got dogs up here, which are doing pretty well. They got somewhere around 800 receptors that are active. The hashed bar, these are pseudogenes. So these are old genes that used to code for receptors, but which have since become genetically broken. Like over time, they developed a mutation in these genes, they no longer worked, and the animal still survived. And so, yeah, it just goes on carrying these dead genes. There's yeah, a lot of pseudogenes for odorant receptors, for sure. And that's fine. It just means these don't make any particular contribution to the current survival of the animal. The animal survives fine without it. But yeah, look at that. Dogs have pretty good sense of smell, but look at rats or opossums. Oh my goodness. Or even cows, yeah. I mean, this would suggest that police should have blood cows rather than bloodhounds, maybe, or just a little rat on a string that they say, "Yeah, go out, find the scent, fetch him." And they certainly have a lot of odorant receptors, and so potentially can smell a wider variety of smells than you or I, which is very interesting, I think. Okay, so these are this is the tree of all of the receptors that are known on humans. And so the branches of different colors are showing which ones are related to each other. The red branches are the ones where we actually know what the chemical is that stimulates them. So get a load of that. I don't know if that's 5%. It doesn't even look like 10%. But yeah, we have a very small range. We know what binds to a few of these odorant receptors, but for most of them, we do not know. So all these other receptors are things that are in your nose being expressed right now, and we're not sure exactly what they bind, but some of those you can smell a rose and you will smell it. Jasmonic acid or whatever it is will come through to you. And same thing with every spice, same thing with everything, like wet leaves, lawn clippings, whatever it is, you have one or more, it could be a group of these receptors responding to different chemicals. And by turning on multiple receptors at the same time, you're going to create some sort of basically electrical signal inside the brain, which is going to be perceived as some kind of thought, which may be connected to some kind of memory, and which is, eventually going to be hooked up to your consciousness. So smell, just like vision, is another very direct way of getting at the consciousness, seeing what's in there, and possibly even changing what's going on in there. So yeah, all that scented candle nonsense, I don't know. It's it's certainly not science, but at the same time, smelling things that you like may make you feel a little bit happier, and yeah, I don't know. That's probably real. Okay, so where do you find all these odorant receptors? There are a couple hundred, let's see, we've got 630 odorant receptor genes in the human chromosomes. And you can see some of the areas where these things are distributed. So each of these is a chromosome. The little stripy bar in the center is the chromosome. And you can see that pretty much every chromosome except the Y chromosome is going to have odorant receptors that appear to be active. It looks like chromosome 20 does not, but chromosome 8, P23 definitely does. They just haven't marked it on there, and so I put that in there. So yeah, odorant receptors pretty much all over your human genome. All right, so let's talk about some interesting stuff. Yeah, so this is a really neat study where they are looking at sensitivity and the change in sensitivity to a particular chemical called benzaldehyde. And this chemical really could have been anything, but they tested for sensitivity to smelling this. And so they had different little like cotton swabs that had different amounts of this chemical on them, and they'd keep them, they'd make a new one, they'd have it in a little jar, and then they'd pull it out and they'd say, okay, can you smell it or not? And people would say, yes, I can smell it. And then at a certain point there wouldn't be enough, and they would say, no, can't smell it anymore. And so they would record, okay, what point is it where people can no longer smell the little Q-tip or cotton swab or whatever it is. And look at the differences here. For males, they did this over many test sessions, 30 different test sessions. And as you can see over time, we got a little bit of a sensitization, just a little tiny bit. At least in some males, there was they were a little bit more able to make to smell this later than earlier. So they developed a little bit of olfactory memory, smell that kind of locked onto something, and they could remember and then smell it at lower and lower amounts. Look, though, for the genetic females in the study. Look at that. By the time they were done, they're smelling about 10 to the minus 5 millimolar, whereas at the start, they needed about 10 to the minus 1, so it's a 10 to the minus 4 difference. 10 to the 4th is 10,000-fold. So over the course of 30 repetitions of this thing, the women in the study ended up with 10,000 times. They could smell the thing when there was 1/10,000th of the amount that originally they smelled it. So I don't know if this jives with your experience or not, but it may well be that you know somebody who's grown more and more intolerant of smell over time. Or just, it could be in a good way or in a bad way. But this one, wow. It is strong and it is real. Now, this doesn't happen to everybody, and this doesn't happen all the time. They tried the same experiment with taste, and this was in men and women, and you can see over the course of the experiment, they both got a tiny bit more sensitive, but just like the tiniest bit. It's essentially a flat line, and no real difference. So for certain smells, potentially certain sets of gene expression. So it's nothing necessarily about having 2X chromosomes. It's about everything that those have done for an entire lifetime, all the ways they've set up the different cells, the amounts of different proteins that they have stored up because of these 2X chromosomes over a lifetime, and because of the lack of a Y chromosome. That's where this appears to come from, and it can't be any more certain about that at this point, but thought it was a kind of a neat little study. All right, another one. Odor affects happiness. Yeah, you bet it does. Yeah. So this is a very kind of sad study. they took patients, this is a survey, and so they had them rate the odor of their partners versus various other people. And so I think this was all done with heterosexual, males and females. And so they had them smell the females would smell other males, the males would smell other females. They'd just have them, for these sort of tests, you would normally tape like a cotton swab under the armpits, wear it around for a day with no soap and no deodorant, no nothing, and get that just loaded up with smell, seal it for freshness in a nice Ziploc bag, and then, yeah, they cut this up into little pieces and use it for different people. So, what this is showing is that from here's very unpleasant, neutral, pleasant, or very pleasant. This is how people are rating their partner's odor. There are. So depression is on this graph, and look, depression is highest in the people who had unpleasant smelling partners, and is actually quite lower when you like the smell of whoever your partner is. So yeah, same thing with an anxiety score. Yeah, much higher levels and statistically significant than in people who had what they considered to be pleasant smelling partners. Now, I don't know that there's a particular compound that makes a partner smell pleasant. I think it's probably going to be a combination of everything that you have learned in your entire lifetime and the way your nose and brain and entire neural structure is built and what genes you have. But definitely this would say, before you hook up long term, give your partner a little sniff, and that may help decide a little bit of your future happiness. All right. Now, there is a pretty strong relationship between scent and memory in older adults. And again, I thought this was a kind of a neat little study. What they're doing here is just testing for memories. They'd give them a smell of something like, cookies or something like that, different things. And they had a way of measuring, like how many things can you remember associated with this smell. And so they'd give you, 5, 6, 7 memories, whatever. And the neat thing here is that for the older people in this study, so they had moderately strong memories of just, things they'd smelled recently. Not very strong memories of everything they'd smelled in the middle, but look at that toward childhood. Some of these childhood smells actually were still really embedded in their consciousness, and triggered very strong memories, which I just think is kind of nice. Even when you're old, there's a little person, there's that little child still there inside of them. All right, but there's a dark side to this as well. This is from a thing called the peanut butter test, which I've seen some studies that say that this needs to be revised, but it worked pretty well in this particular instance, and I think there is something here. Maybe not the sidedness, but something. So the test is they had people with, so first they checked, make sure their nostrils are both open. They can breathe normally through those and smell things, or just breathe normally through those. And then they had a ruler and a little cup of peanut butter, 14 grams of peanut butter. And so they would put the peanut butter, they'd have the person blindfolded, and they'd have them close one nostril, so they're only smelling through one nostril at a time. And they would move the peanut butter closer in sort of 1 centimeter increments until, during the exhale, until their inhale, they could say, oh, I smell it, that's peanut butter. And the neatest thing was that they found really strong, one-sided smell in Alzheimer's disease patients. And they also found fairly strong one-sided smell. It was on the other side in this case, but in other kinds of dementia patients. So for whatever reason, there was a strong bias toward the left nostril was much better than the right nostril in Alzheimer's disease in this study, and the right was better than the left in other kinds of dementia. In the older patient controls, which are right here, you can see they were right in the middle. They can smell equally at an equal distance. It's just, And so what this is pointing out, I think, is that there's actual damage, particularly between in different parts of the brain and perhaps in communication between different sides of the brain that you find in Alzheimer's disease and other dementias. And you can show it up with little tests like this one, this peanut butter test. Here's another bad one. So this is an odor map. They took Alzheimer's patients and people who were the same age but did not have Alzheimer's, and they had them rank different smells. There were smells of like fruits and things you'd find around the house. And they had them rank them from pleasant to unpleasant, edible to non-edible, fruity to non-fruity, and a bunch of other sort of scales. And then they're showing all of the most significant data. And so for the healthy controls, which are up here on top, the elderly controls, you can see that they think that coffee and chocolate and cinnamon, these came out as fairly pleasant. Yeah. And vinegar is very unpleasant. I totally agree with that. I cannot stand the smell of vinegar. It just makes me mad. And yeah, the old people have got me on this. They could totally back me up. And they were able to identify some other things as baby powder was kind of pleasant, but mostly it's skewed toward non-edible. And like nail polish remover definitely smelled non-edible, but pineapple, orange, banana, those all seemed like things that you could probably eat. Yeah, based on the smell, yeah. But look down here in Alzheimer's disease patients, and what you see is the clusters are all broken up, and you've got some weird stuff. So the Alzheimer's disease patients weren't actually able to generate significant associations between edible and non-edible. They were just kind of all over the place. The best they could get was to say that baby powder was kind of pleasant and fruity, but they may not have been able to pinpoint exactly what it was. And like some of the fruits, banana, pineapple, orange, they are being classified as fruity. Yeah. And so is vinegar, actually, a little bit, although it's still unpleasant. And I think that's also very nice that the unpleasantness of vinegar is sort of hardwired into the brain. But yeah, basically unable to tell what's edible from what's not edible. They can tell that coffee and cinnamon don't smell fruity, but yeah, had no idea about edibility. So that was really kind of sad. But this is just showing once again that when something changes in your brain, your senses will also change. Your sense of smell in particular is kind of a gateway into what is happening inside the brain at any one time. Yeah, scent is definitely going to influence perception. So if we were there in class, I've got this great little bottle. It is it's called Hogmate, maybe Hogmate Plus, I can't remember. And it is a pure swine pheromone, which is just the same, in this case, it's a dihydrotestosterone, if I remember right. Oh, actually, no, it's an androstenone. There we go. And so this is a very male sort of hormone. And this is a potential thing you can use to induce mating in your pigs. Apparently, most of the people in the Amazon comments section were talking about spraying it on cats to get them to stop mating with each other or harassing each other. So, I don't know. Anyway, yeah, so this is showing an interesting thing. So if we were to go to this cell paper, it's got a little animation in there. And the animation is going to show you what looks like people that are walking. And they're just going to show you the key points, the hips, the shoulders, the arms, and the legs and torso. So it's just, maybe 10 little spots that make up a person. And they have these ranked from a very masculine walk to a very feminine walk. And I'll make sure the link is there in the... actually, no, the link is right here. So you just click that link and go and watch it. And when you watch this, if you are smelling female hormones, it will actually bias heterosexual males to make that pattern of dots appear more female and less male. And likewise, if heterosexual females are smelling the male hormone, it will actually bias them to take the same ordinary set of dots. So there's one right in the middle of this, and perceive it as being a little bit more male. This is the olfactory equivalent of beer goggles, I guess. And so let's actually come out of this. Let's open up this particular link, see if we can get this to run. And if this does not run, then we will, after Futurama anyway, we will go, and there we go. Okay, we got it. So what you got here are, yeah, various degrees. So let's watch. Watch this one here on the end. Okay, you see what the hips are doing? So this hip sway there, this is modeled on actual females. They had females walk. And so what you got here is about the most female that they've got, or rather a 0.45 on the scale. That's what they considered reasonable. And then contrast that rather to this side. Now look what's going on with the dots on the right. Look at those hips. See how those hips are just solid? That for me is like Dwayne "The Rock" Johnson walking into a room. Just, yeah, picture that. Picture, I don't know, whoever you want that's female on the other side. And yeah, can you see it? I think you can see it. Yeah. All right. And so what they did was they took all these little samples of different, sets of dots, and then had people say, okay, is this more male, more female? Where would you put it on the scale? And they found that there was a difference. They would see the same set of dots as more or less masculine or feminine, depending on what they were smelling at the time. So I think that's just fascinating. Okay, and here's another one. This is a interesting thing to bring up, yeah, when people are talking about this sort of stuff. So this is a test looking at heterosexuality versus homosexuality and the influence of odors. And what you've got here are, so this is one of those studies where they had people wear around the little patches, and they had no perfume, no whatever. They had to be, I think, starved of soap and perfume and washing with anything other than water for a couple days. And then they attached these cotton pads underneath their armpits, wore them around for a normal day, a normal, somewhat smelly day, I would imagine. And then, yeah, sent these off to the lab for analysis. And they took heterosexual males, gay males, heterosexual females, and lesbian females, and gave them each these things to smell and had them rate the smells on a scale from, fairly pleasant to fairly unpleasant. And so here are the results. The first thing I'll point out is that you notice nearly all of the bars point down. Basically, everybody smells bad when you do a test like this. I think that's just, maybe what you would expect. But not everybody smells as bad as everyone else. And so for heterosexual males, look at that. The pads from gay males were ranked as much more unpleasant than the ones from, for example, heterosexual females or other heterosexual males. Yeah, interesting. And given that there's, if homosexuality is only a decision, then what the heck is activating the odorant receptors of a person differently? Because this person's being given different pads from different people. And this is by far not the only study. There are many of these out there that generally replicate the findings of this one. This is just kind of a big early one that I thought was nicely presented. Yeah, same thing over here. So gay males rank heterosexual males as smelling fairly bad, but other gay males, there's a decent error bar on there, but there is a significant difference between those two. And so, yeah, gaydar, if it is real, may be olfactory. Similar thing for heterosexual females. Did not like the smell of heterosexual males, gay males, particularly compared to anybody. I mean, didn't like any of the smells, really. But yeah, thought heterosexual males were maybe not so bad. And homosexual females also now actually thought that heterosexual females smelled okay, but other homosexual females were also fairly okay. So, yeah. So to some extent, most likely, your choice or whatever, your orientation is probably hooked up to other stuff in your brain. And this stuff seems to be hooked up to the odorant receptors and these pathways. Now, it may not be the molecule itself. It may not be that odorant receptor itself, but some of the ways that it's hooked up in the brain and all the other things going on around it. Remember, we're taking a very complicated cell and trying to simplify it down to just, what's this one protein doing? When that is never really the case in biology. It's always a complicated stew of swirling proteins, each going about their different activities and being made and broken down at any one time. And so that's why, people are tough to predict, or anything living is kind of tough to predict. Okay. This was another study that ended up showing. Actually, no, this is another thing from the same study. This is just showing with the size of the circles, basically preference and which group preferred the smell of which other group, or didn't necessarily mind the smell of which other group. And in general, mate choice was related to olfaction, even though in these studies there was no clue as to whether the scent patches would have come from male or female or anything else. Basically, it's just molecules on this little cotton pad, and the molecules are acting differently in different people's brains, which I think is actually kind of neat. Yeah, so there you go. All right. This is actually one of the receptors that they think may be involved in this process. So odorant receptor 7D4 is the olfactory receptor for androstenone. Androstenone is one of the, it's a male hormone. If you go to a really nasty public bathroom that has a urinal, you will notice it's kind of gross, but also kind of sweet smell on the area around the floor if they don't clean it very well. That is kind of what an androstenone smell is like. I don't know, it burns and I absolutely hate it, but yeah. It's there, and that's what is in this Hogmate stuff, which incidentally, they do sell male and female hormones. You can buy these things in sprays online, and boy, they smell awful from what I can figure. But anyway, this was from a really nice paper that looked at odorant receptor number 7, D4, and they figured out, first of all, that this one was selectively stimulated with androstenone, which here we go, and it's going to be one of these steroids related to testosterone. You see the two little methyl groups sticking up in the air. And androstadienone, yeah, we said that right, which is again related to testosterone. You got the two little molecules sticking up there. And so, yeah, and the double bonded oxygen for extreme double bonding down here. There you go. So, yeah, they exposed these people, or rather they had cells just in a dish, exposed them to all these different chemicals, and found out that these chemicals made the cells start doing something. It induced a change inside the cells. It was a signaling change. And that all the other cell scents that they put into that cell culture did not. So that kind of shows that, yep, these are the ones. They seem to be linked. And then interestingly, they looked in people, and they found that a lot of people had the wild type version of this, which is called the RT version. But they found some people that had the RTWM version. And the point of the WM version is that it can smell these things, but it smells them much less. The RT version smells them very strongly. And remember that a strong scent doesn't mean necessarily a pleasant or unpleasant scent. Think like laundry detergent, laundry freshener. It's pretty darn strong, but it's meant to be pleasant. Yeah, and then think, I don't know, cat pee or something. Yeah, very strong, very unpleasant. Yeah. But they're both strong, and they're coming through different receptors. So, yeah, there's a sort of strong scent phenotype, and there's a weaker scent phenotype, the RTWM heterozygotes. And there will be some people who'd be WM, but those are somewhat rare. I think they didn't have enough in this study. And they found that in these people, there was a big difference. They didn't really perceive many differences in the intensity of these other smells. They could still smell them at the same concentration. But there was a big difference in the RTWM. The androstadienone and androstenone were both much less intense than they were in the RT, the people with the wild type version. Yeah, and they were also found to be, because they were less intense in this case, they were also correlated with being more pleasant. So you would say, oh, people with RT-WM might be inclined to perceive androstenone as somewhat more pleasant than people with RT genetics. And again, this is probably not a thing. I don't think there really is anything like a gay gene. I know there have been people talking about it for a while, but this may be a little part of the puzzle. And this is a genetic difference that could be inherited that would potentially link up with perception of scent from different people and could potentially influence mate choice. Although, most people are going to be RT, and so they are going to perceive the world in just about the same way. And yeah, we're all here, so somebody was getting together, despite not having the RT-WM phenotype. The other interesting thing is that you do get RT, and it's a very strong signal. WM gives almost no signal down here. It's kind of buried along the bottom. You can barely even see it. There are other variants of this out there. So there's a rare variant called S84. This gives extremely strong, so it's a higher line, so it's extremely strong olfaction of androstenone. And there's a super special variant that you only find in chimpanzees, the chimpanzee variant. And this one is extremely, extremely strong. Look at that thing. So there are different levels of scent perception involving androstenone. And chimpanzees are sometimes known to physically tear apart male chimpanzees that have acted out of order. Sometimes the females will apparently get together and just, rip them limb from limb. And so I don't know if this particular odorant receptor, this chimpanzee version of odorant receptor 7D4 is going to be involved in there. But somewhere in chimpanzees, there exists a receptor that is much, much, much more sensitive to, to male scent, basically, than in other creatures, such as ourselves. So, interesting. And differences like this almost have to play out in some way in the world. And it's not the sort of thing that you would ever know. I don't actually know what genes I have, and you probably don't know either. You could sequence them. You could figure out which version of the odorant receptor you had with, a little PCR, which is something we're going to cover in a couple of weeks, and a couple of primers. And yeah, there's a chance that this would explain something about who you are. But remember that it's not just genes, it's also how you hook the genes up and what memories you've laid down, and an entire lifetime of learning that's behind this. So yeah, that's something. Anyway, this is just hammering home the idea. This is a nice little rat brain, and they have a big old bulb up here. This is called the olfactory bulb, and there's actually an accessory olfactory bulb with some of the cell bodies in it. And each of the cells in here is going to be expressing different odorant receptors. So just like we had rods and cones that can express different genes, different G protein coupled receptors like rhodopsin that will help you see different colors, there are also going to be cells that express different profiles of odorant receptors. And so in the nose, you're going to have this neuroepithelium that's going to have all these different cells. Each one's going to be connected up, because this is a neuroepithelium. They're going to be connected up to the olfactory bulb neurons. And then from the olfactory bulb, we're going to process that smell in some way and eventually turn that into some kind of conscious thought, or at least unconscious revulsion, right? Yeah, when you smell certain things. And it's not just an individual receptor going off, because most scents are going to be complex outside of the laboratory, in a test where you only have one scent or nothing. Most scents are going to be a mixture of different things that are coming off the same organism or situation or substance. And so you're going to light up a bunch of different receptors to different amounts. So if you can imagine different odorants here, they may all hit the same receptors, but one may hit really hard on receptor number 3, a little bit on receptor number 1, and nothing on 2, 4, and 5. And that particular profile of scent reception is what's going to be transmitted as thought inside your brain. And your brain has built up. It's a really cool computer, and it's built up ways to actually understand, kind of make sense out of scent. Yeah, and if we don't fully understand that, to understand that would be to kind of understand consciousness itself. But people are working on that. All right, and the last little thing before we go into the mechanism, which I'm going to do as a separate little slideshow. So this is looking at certain kinds of birds, if I remember right. Yeah, what you can see here is the olfactory bulb on these, and so it's the blue part that's up here. And so in some of these types of birds, you will find a very strongly developed olfactory bulb, and in others, the olfactory bulb is really small. And we think this reflects basically usage patterns. So in birds where they are very strongly scent determined, where scent plays a big role in mating or in finding food or finding your way to your house or something like that, then there is going to be a strong olfactory bulb to perceive this scent because it's going to be hooked up. It's going to be a major part of thought. And in the other birds where scent plays essentially no real role, you're going to find that, yeah, the olfactory bulb may be really tiny, kind of shrunken, if you like. So there we go. Interesting little thing. Okay, and then we'll go into the mechanism in just a moment. But that is the end of this part, which was just to give you kind of a frame for some interesting connections between the olfactory senses and the rest of the brain.